The last episode, I was talking about passive mechanisms of movement across the plasma membrane. Sometimes the molecules need help across the membrane, and so they use proteins as carriers or as channels to move across the membrane. This can be either passive or active. So here we have in this area here, you can see facilitative diffusion. In facilitative diffusion, molecules move from higher to lower concentration down their concentration gradient, but they need a facilitator. And so that's why it's called facilitative diffusion. It's still just a passive mechanism, but we're using a protein channel. Active transport mechanisms are going to either move molecules against their concentration gradient, or they need energy to move the molecules across the membrane. So here you're looking at Here you're looking at active transport, moving molecules against their concentration gradient from lower to higher concentration. So you see these little tiny boxes. These are some type of molecule, and they're moving against their concentration gradient. They're using the energy of ions moving down their concentration gradient, so from higher to lower. So the hydrogen ions are moving via facilitative diffusion, while these boxes are moving against the concentration gradient. That's how we transport glucose into cells a lot of times. Same with bacteria. We can also bind molecules, and once bound, the molecules are able to move down their concentration gradient. Or we can use translocation, where we're altering the molecule slightly to allow it to move across the membrane. So this is active transport via group translocation.
Group translocation is a process in which an organic molecule, such as glucose, is transported into the cell while being chemically modified. The PEP phosphotransferase system is an example of group translocation. In this process, the phosphate from the high-energy compound phosphoenolpyruvate, or PEP, is transferred to HPR by enzyme 1, and the product pyruvate is released. The phosphate is then transferred from HPR to enzyme 2B by enzyme 2A. The membrane-bound enzyme 2C then carries the glucose across the membrane, and the glucose becomes phosphorylated as it enters the cell.
And so we've modified that glucose molecule as it's moving across. Another mechanism of transfer, but also a mechanism of producing ATP in that plasma membrane, is called the electron transport chain. And so in the electron transport chain, which you've all probably heard of in either your general biology class or in anatomy and physiology, is a mechanism by which we use electron transporters to pump hydrogen ions across a membrane from lower to higher concentration. And as electrons are shuffled from one proton carrier or from one electron carrier to the next, hydrogen gets pumped across the membrane. As the hydrogen fly back through to the other side from higher to lower concentration, they produce ATP. So this is a mechanism in which we produce the majority of the ATP that organisms use. And here is your proton pump.
Proton pumps are protein complexes that move the protons generated during oxidation reactions across the cell membrane. As the protons move through the proton pump, they begin to build up on the outside of the membrane. The protons accumulate on the outside of the membrane, creating a concentration gradient. The membrane is not permeable to the charged hydrogen ions, and they cannot diffuse back across the membrane. Instead, they must pass through a special channel. The protons move through this special channel, which is the enzyme ATP synthase. This enzyme uses the energy derived from the movement of these protons to convert ADP into ATP. The movement of protons down a concentration gradient provides the energy for ATP synthase to form ATP. This mechanism of producing ATP is called chemiosmosis.
All right. We'll be talking more about that when we get into metabolism. So now we're going to switch gears and we're going to talk about the cell wall of bacteria. Bacterial cell walls are very strong, and in general, all bacteria contain a cell wall. One of the most distinguishing features of the cell wall of bacteria is the amount of peptidoglycan found within the cell wall. Gram-positive bacteria contain a lot or multiple layers of peptidoglycan, while gram-negative bacteria contain very, very little peptidoglycan. And this difference actually allows us to identify gram-positive bacteria from gram-negative bacterial cells, because gram-positive bacteria stain purple in a process called the gram stain, while gram-negative cells stain pink. There are a lot of other differences between gram-positive and gram-negative cells, and you can see them here. Thick versus thin layer, teichoic acid is present or absent. Gram-negatives don't have the thick peptidoglycan, so they have an outer plasma membrane, so they have two plasma membranes. They also have a lipopolysaccharide found within their outer plasma membrane. We'll talk about some of these in future sections, okay? So peptidoglycan is a protein carbohydrate layer. And you can see what peptidoglycan kind of looks like here. Peptidoglycan in gram-positive bacteria, there are about 30 or more layers, whereas in gram-negatives, there's only a few layers. They also contain teichoic acid within the peptidoglycan, and this gives the gram-positive bacteria the negative charge that allows it to stain with those basic dyes. Peptidoglycan is actually produced during cell replication, and penicillin inhibits the production of peptidoglycan, and because of this, gram-positive bacteria are very sensitive to penicillin. Penicillin binds to the cell wall and inhibits peptidoglycan synthesis, which is why it's more effective against gram-positive than gram-negative bacteria. So this is a cute joke. I love this. I found this on Google Images. I was looking up something cute about peptidoglycan. This is called The Bold and the Bacterial. I don't care if he is penicillin, we're in love. So gram-positive bacteria talking to his parents or her parents, and in love with penicillin. Kind of cute. So what happens, the destruction or the inability to produce peptidoglycan doesn't cause the bacterial cell to die. But instead, what it will do is it will inhibit the cell wall formation. And the cell wall is what protects bacterial cells from osmotic lysis. Lysozyme is another chemical that can cause osmotic lysis because it breaks through those peptidoglycan layers, and when it does so, it causes the cell wall not to have the integrity that it did, and so the bacteria are more prone to fluids moving in, causing the cell to burst. So here is a bacterial cell. When you add penicillin, the cell wall won't form. So if that was in an isotonic condition, nothing would happen to the cell. But when you move that cell into the hypotonic solution, fluids will move in, causing the cell to swell and eventually burst open. So here's another figure showing gram-positive bacteria. They have one plasma membrane. They have a thick layer of peptidoglycan outside of that plasma membrane found within the cell wall. Gram-negative bacteria have very little peptidoglycan. They need something else to protect them. What they have is a second plasma membrane. So they have an inner and an outer plasma membrane. And within the outer plasma membrane, they have these lipopolysaccharides that are attached. These lipopolysaccharides are really important because they contain two major structures. They contain a lipid called lipid A and an O antigen that identifies the different bacterial species or strains of bacterial species. So when you think of the lipopolysaccharide, the lipid A is actually an endotoxin. And when it gets into our blood, it's going to travel to our hypothalamus. This endotoxin acts as a pyrogen. And if you don't know what a pyrogen is, it's something that causes heat production. So think of a pyromaniac as someone who likes to play with fires. Pyrogens are something that causes fires or causes heat. Lipid A moves to our hypothalamus and stimulates our hypothalamus to set our body temperature at a higher rate. The hypothalamus controls body temperature, but in the presence of lipid A, it's going to increase that body temperature. And that can lead to endotoxic shock, where we have such a high temperature that we can't survive. The O polysaccharide chain, then, is what we use to identify different strains within a single species. So I always like to use dogs as examples. You can have lots of types of poodles. They're all poodles, but you can have long-haired poodles, short-haired poodles, teacup poodles, tiny poodles. I don't know. I'm sure there's other types as well. But they're all poodles. With bacteria, you can have different types as well. So I use E. coli. It's like the most common bacteria most people know or have heard of it. E. coli lives in our gut. It's really beneficial. It helps us to produce the vitamins we need for blood cell production. So without E. coli, we wouldn't be able to produce blood properly. It helps us in breaking down foods. It's part of our normal flora. And so if we didn't have it, we would be sick. But there's also E. coli that can cause disease. They can cause diseases like enterohemorrhagic colitis, which was in one of the disease assignments. So the E. coli that does that is different from the E. coli that lives in our gut. And we can identify them by this O polysaccharide chain. So E. coli 0157H7 is the pathogenic strain of E. coli that causes lots of problems with our gut. And then I told you the lipid A is the structure that we call a pyrogen that enters the blood and upon the death of bacterial cells, gram-negative bacterial cells, and it causes our temperature to increase. So when you have an infection with gram-negative bacteria, a lot of times the infection is going to get worse before it gets better because you're killing off all those cells, which is releasing a lot of these lipids, which are all pyrogens, which will all go to the hypothalamus and all cause you to increase your body temperature. So I'm going to stop here, and I'll start here on the next lecture.